Welcome to Sunnyside. Video number 2 on lipid. Topic: lipid in circulation. Finger-like projections called villi. These line the interior wall of the small intestine. Within the villi are blood vessels that function to absorb digested nutrients from enterocytes. Other vessels present within the villi are the lymph vessels called lacteals or lymphatic capillaries. Blood vessels in the villi absorb most nutrients from enterocytes, but lipids and fat-soluble vitamins are absorbed by lacteals before reaching circulation via the lymphatic system. In the enterocyte, monoglycerides and diglyceride from dietary lipids combine with a polypoprotein B48 to form chylomicrons, a large triglyceride-rich lipoprotein. Like other lipoproteins, chylomicrons carry esterified cholesterol and phospholipids. During intestinal absorption of fats and fat-soluble vitamins, lacteals become filled with milky white lymph called chyle. From lacteals, chyle travels via the lymphatic system into blood circulation. We shall now be looking at activities of chylomicron in blood circulation. So, in overview, we can say chylomicrons are large triglyceride-rich particles known as lipoprotein. It is a composition of lipid and protein, and it is synthesized in enterocytes or intestinal cells. It functions to transport dietary lipids to peripheral tissues and hepatic system. There are other types of lipoproteins. Each has its own characteristic protein and lipid composition. These other lipoprotein types, namely VLDL, LDL and HDL, are synthesized in the hepatic system. In blood circulation, chylomicron lipoproteins, together with other lipoproteins, are hydrolyzed by lipoprotein lipase activated by a polypoprotein C2. Hydrolysis of chylomicron or other types of lipoproteins releases its components of free fatty acids, glycerol, and lipoprotein remnant particles. Free fatty acids are also derivable from adipose tissues' lipolysis of triglyceride. They are then taken up by tissue cells, for example, muscles, liver, and adipose tissue. Our primary energy source is carbohydrate, but during fasting or low blood glucose, we have free fatty acids as a secondary energy source. So, free fatty acid enters cell cytosol and undergoes a series of chemical reactions that, amongst all, includes fatty acyl-CoA synthetase, carnitine acyltransferase type 1 and type 2, and then beta oxidation. The resultant molecule in the name of acetyl-CoA joins Krebs cycle to produce FADG2 and NADH that enter electron transport chain, leading to oxidative phosphorylation for more ATP or energy store production. In this refresher of note on lipid, we have seen that not all body lipids are dietary. Other lipid sources are via specific biological processes. For example, 1. Mevalonate pathways leading to cholesterol synthesis. It is a synthetic process regulated by feedback mechanisms and concerned enzymes. 2. Allosteric or hormonal regulated fatty acid synthesis. In subsequent lessons on lipids, we shall be looking at the benefits of lipids, diseases caused by lipids, and applicable medicines.